climes. This is an example of one of those thermal climes where we have latitude on the y-axis and mean reflectance plasticity, which is a response to temperature, on the x-axis. And of these 29 European populations of Plantago lanceolata, there was a significant latitude by plasticity correlation, where populations found more in the north had much higher average plasticity than populations found further in the south. So besides in Plantago lanceolata, we often see this pattern in many organisms. And it seems to be adaptive because in these northern cooler climates, there's often shorter growing seasons, and the reverse in the warmer climates. So many studies have been published showing these climes, and they seem to be adaptive. However, there are other factors that may have caused these climes. For example, if individuals from the south were derived from a different common ancestor than individuals in the north, we might also see these patterns. So by looking at neutral genetic divergence, we can see if neutral factors can explain these patterns, and therefore evaluate the adaptive value of plasticity. My second question is, how are plasticity genes inherited? And I've asked this question because the mode of inheritance, whether these genes are coming from nuclear or cytoplasmic inheritance, can influence selection. For example, in nuclear genetic inheritance, the rate and direction of selection, the rate and direction of the response are directly related to the magnitude and direction of selection. So for example, this population with some median thermal tolerance, under our climate warming, we might expect to see individuals with a small thermal tolerance die off, producing a population of survivors with a higher mean thermal tolerance. And after one generation of selection, that population can evolve to have an increased thermal tolerance. However, cytoplasmic gene inheritance can cause time lags or continued evolutionary change after the selective pressure ends. My third question is, what is the genetic architecture of thermal plasticity? And by genetic architecture, I'm talking specifically about the number of genes and the interactions between those genes. There have been 3 genetic models proposed to explain thermal plasticity. The first is the overdominance model, in which plasticity is a function of heterozygosity. The second is the allelic sensitivity model, in which allelic expression is sensitive to the environment. And the third is the epistasis model, in which there are 2 sets of genes. Regulatory genes that are sensitive to the environment control the expression of trait-determining genes. Each of these models has different evolutionary implications as well. So in the overdominance model, because plasticity is a function of heterozygosity, we might expect small populations that have increased homozygosity due to inbreeding be unable to respond to selection for plasticity. If the allelic sensitivity model underlies thermal plasticity, then we might expect it to evolve through introduction of new alleles, which could occur through migration or mutation. And if epistasis underlies thermal plasticity, then selection for those regulatory genes may also cause a correlated response in other traits. So far, empirical research hasn't really found much evidence for support of the overdominance model controlling plasticity. However, many studies have supported the allelic sensitivity and epistasis models. And my fourth research question is, what are the genetic correlations between the thermal plasticities of different traits? Genetic correlations can arise due to physical linkage between genes causing them to be inherited together, or by pleiotropy, in which one gene influences multiple traits. This can be adaptive if both of the traits, the selected trait and the correlated trait, increase fitness in the new environment. However, if the selected trait increases fitness in the new environment, but the unselected correlated trait decreases fitness in the environment, this is called antagonistic pleiotropy, and it can halt or alter the response to selection. So genetic correlations, selection for one trait causes a correlated response in some unselected trait. Oftentimes, genetic correlations are observed in a single environment. However, few studies have observed correlations in multiple environments. One of those studies found that in Arabidopsis thaliana, individuals that delay their flowering in response to cool temperatures had more leaves prior to flowering. So you can see how this might be adaptive in some environments if having more leaves before you flower allows you to produce more flowers and more seeds, thus increasing fitness. So these are my 4 research questions, and I chose them because, again, a major goal of biologists is to forecast and mitigate the effects of climate change. By answering this first question, we will be able to determine the adaptive value of thermoplasticity and identify in which environments it may be adaptive. These other 3 questions will allow us to better predict the future response of plasticity to selection, especially in a warmer climate. And now on to Plantago lanceolata. Plantago lanceolata is a wind-pollinated perennial herb that's widespread around the world and is native to Eurasia. It has a gametic genome, or it has about 1 to 1.3 billion base pairs in the genome, and those are arranged onto 6 linkage groups. I chose Plantago lanceolata to study thermoplasticity for many reasons. The first is that under short day conditions, it grows as a rosette, and it will continuously grow as a rosette, producing more leaves under short day conditions. During this time, it can easily be sliced through the apical and root meristematic tissue into multiple clones, which can then be introduced into multiple environments. By introducing it to long day conditions, flowering is induced, in which these long leafless scapes emerge from the leaf axils, on the top of which are flowering spikes. And these flowering spikes consist of a tightly packed cluster of flowers. These begin flowering at the proximal end and proceed distally, with the female parts emerging first, followed by the male flowering parts. Thermoplasticity in these flowering spikes has been studied extensively by Dr. Lacey. And we found that most individuals, when grown in warm environments, produce these lightly colored, highly reflective spikes. Here I'm showing floral reflectance at 850 nanometer wavelength, grown under cool and warm temperatures. So some individuals, when we grow them in cool temperatures, are unable to change their flower reflectance, producing the same type of spike. However, some individuals can reduce reflectance, and others can do so greatly, producing a much darker spike. When both the dark and light spikes from the same individual are grown in the same environment, these dark spikes are often about 1 degree Celsius warmer, which can allow these spikes to partially thermoregulate their developing offspring and seeds. This is a spectral scan, and it shows how we measure that floral reflectance. So here I have reflectance on the y-axis and our wavelength in nanometers. And you can see that there's a lot of overlap down here at the low wavelengths. So that's why we chose 850 nanometers as our measure of reflectance, because they're much more spread out. What's shown on the graph is data from 3 different genotypes grown at high temperature and the same 3 genotypes grown at low temperature. Under high temperature, all 3 genotypes are overlapping up here because they all produce highly reflective spikes. However, at low temperatures, this NC14 genotype can reduce its reflectance a little bit. This NC19 genotype can reduce its reflectance more. And this NC1 genotype can greatly reduce its reflectance. So this shows that there is genetic variation in thermoplasticity among individuals. However, even though floral reflectance plasticity